BTEC Applied Science Unit 1, A4 Digestion and Excretion Summary. So here we have the excretory system. So there's two kidneys, ureter. The ureter takes the urine to the bladder. The bladder is where the urine is stored, and then the urethra is where the urine is excreted from the body. And here is the structure of one kidney. So you've got, this is the ureter, which you can see here, which takes the urine to the bladder. The blood entering the kidney is called the renal artery. The blood leaving the kidney is called the blood vessel leaving the kidney is called the renal vein. And then these kind of almost circular shapes are called the medulla. And then this bit here, shown in kind of a beigey colour, is the pelvis of the kidney. And then the bit around the outside is called the cortex. And then it's covered in a fibrous capsule. So the kidney capsule is the outside of the kidney, and it is tough and fibrous. The cortex is the outer layer of the kidney, and it has a vast blood supply. The medulla of the kidney is the inner portion of the kidney, and it is composed of collecting tubules that empty into the renal pelvis, which is here. The pelvic pyramid is where the collecting ducts of the nephron drain into from the medulla. The pelvis is the pyramids drain the urine into the hollow funnel-shaped structure, which then leads to the ureter. The renal artery brings blood to the kidney and into the cortex, and the renal vein removes blood from the kidney from the vasa recta capillary network in the kidneys. And then here is a nephron. So there's millions of nephrons in the human kidney. So each kidney contains between 1 and 2 million filtration units called nephrons. So you've got the arteriole from the renal artery, and then the arteriole that comes away from the glomerulus. The glomerulus is just a bundle of capillaries, like a branch network of capillaries, and it's found inside the Bowman's capsule. So the Bowman's capsule just surrounds that network of capillaries. This first bit here is called the proximal convoluted tubule. Then you've got the loop of Henle, shown in yellow, this big loop. Then you've got the distal convoluted tubule here, and then you've got the collecting duct. So the Bowman's capsule, the job of the Bowman's capsule is for ultrafiltration. The proximal convoluted tubule is for reabsorption of water and glucose, which is known as selective reabsorption. The loop of Henle is to maintain the gradient of sodium ions in the medulla. The distal convoluted tubule is to reabsorb water. The collecting duct is again for reabsorption of water. So ultrafiltration that happens between the glomerulus and the Bowman's capsule. So there's a high hydrostatic pressure in the glomerulus, this network of capillaries, because the diameter of the afferent arteriole is a lot wider than the diameter of the efferent arteriole. And the small substances like water, glucose, ions, urea are forced into the glomerular filtrate. Large proteins are too big to fit through, so they will remain in the blood. But all the small things like the water, the glucose, the ions, the urea are forced into the Bowman's capsule by ultrafiltration under pressure. So selective reabsorption then happens in the proximal convoluted tubule. Here all the glucose, amino acids and most of the water and ions are selectively reabsorbed. As the glucose and amino acids are reabsorbed, water will follow by osmosis down the water potential gradient. The cells of the proximal convoluted tubule contain many mitochondria to provide the ATP required for this process. They are also covered in microvilli to increase the surface area to volume ratio for faster reabsorption. Maintaining a gradient of sodium ions in the medulla by the loop of Henle. So the counter current multiplier system. So in the ascending limb, sodium is actively transported out, so the filtrate concentration decreases. But water remains because in the ascending limb, the walls are impermeable to water. And this just increases the concentration of sodium in the medulla, which lowers the water potential of the medulla. And then in the descending limb, water moves out by osmosis because the walls are permeable in the descending limb. So water moves out by osmosis, then it's reabsorbed into the capillaries, so therefore the filtrate concentration increases. And then the sodium is recycled because it diffuses back. Next bit is osmoregulation. Water moves out of the distal convoluted tubule and collecting duct by osmosis down a water potential gradient, controlled by ADH, which increases their permeability. So osmoregulation is the control of water potential of the blood by negative feedback. Roles of the hypothalamus, posterior pituitary and ADH in osmoregulation. So the hypothalamus contains osmoreceptors, which detect the blood water potential, and the hypothalamus also produces ADH when the water potential is low. The posterior pituitary gland secretes ADH into the blood because of the signal from the hypothalamus. The antidiuretic hormone, or ADH, more is secreted when the blood water potential is too low. This increases the permeability of the cells of the collecting duct and distal convoluted tubule to water, so it increases the water reabsorption back into the blood. So therefore it decreases the volume and increases the concentration of the urine. So, how the body responds to decrease in water potential or dehydration, for example, caused by less water intake or increased sweating. So in the hypothalamus, osmoreceptors detect decrease in water potential. So the hypothalamus produces more ADH. The posterior pituitary gland secretes more ADH into the blood. ADH attaches to receptors on the collecting duct and the distal convoluted tubule, increasing the permeability of cells to water. It causes more aquaporins to be put in place. So more water is reabsorbed from the distal convoluted tubule and collecting duct by osmosis. And the urine is a smaller volume, and it's also more concentrated. And then in terms of how the body responds to an increase in water potential, so if you've got over diluted blood, it's just the opposite. So the osmoreceptors will detect that there will be less ADH produced, there'll be decreased permeability, less water will be reabsorbed, therefore there will be a larger volume of dilute urine. Digestion. So here's a picture of the digestive system. We've got the esophagus, the tube that goes from the mouth to the stomach. Here we've got the stomach, the liver, you've got the gallbladder, top part of the small intestine or ileum is called the duodenum. Got the pancreas, small intestine or ileum, large intestine or colon. Here is the appendix, you've got the rectum and the anus. So the stomach is a large muscular sac that continues to the mechanical and chemical digestion of food. The muscular walls churn the food to mechanically digest it, and it contains hydrochloric acid to kill any bacteria ingested, and it's also the optimum pH of pH 2 for protease enzymes which start protein digestion. The pancreas contains both exocrine glands which secrete digestive enzymes like endopeptidase and pancreatic amylase and lipase into the duodenum via the pancreatic duct, And it's also got endocrine glands that secrete insulin and glucagon into the blood to regulate blood glucose levels. The gallbladder stores bile that is produced by the liver, and it releases the bile via the bile duct into the duodenum, and the bile has a pH of 8 to neutralise the stomach acid. It also emulsifies lipids to produce a larger surface area for lipase to act on. The duodenum is the first part of the small intestine, or ileum, and it is the site of neutralisation of stomach acid using the bile from the gallbladder. The ileum is part of the small intestine. It has microvilli on the epithelial cells for the site of absorption of the products of digestion, which then move into the bloodstream. So glucose and amino acids, either by facilitated diffusion or active transport using co-transport, or the lymphatic system, the digestive products of lipids move by chylomicrons and then are removed. And then the colon is the site of water reabsorption, or large intestine. So starch digestion starts with salivary amylase, which hydrolyzes starch into maltose. Pancreatic amylase can be secreted into the small intestine, and the brush border enzymes, so the membrane-bound disaccharidases, can be found in the lining of the epithelial cells that line the small intestine, and they can secrete some disaccharidases called like maltase, which hydrolyzes maltose into glucose. So carbohydrate digestion, it starts in the mouth with amylase, which hydrolyzes starch into maltose, and then in the duodenum, pancreatic amylase continues to hydrolyze the glycosidic bonds in starch to maltose. Maltose is then hydrolyzed to glucose by maltase enzymes embedded in the membrane of the epithelial cells lining the ileum. So, summary, name of the enzyme amylase is produced in the salivary glands and pancreas, and it catalyzes the reaction of starch being hydrolyzed into maltose. Maltase is embedded in the epithelial cell membrane of the ileum, and it catalyzes the hydrolysis of maltose to glucose. Absorption of glucose now. So sodium ions are actively transported out of the epithelial cells lining the ileum into the blood by the sodium-potassium pump. This creates a concentration gradient of sodium, a higher concentration of sodium in the lumen than the epithelial cell. As the sodium ions are carried into the epithelial cell via a co-transporter, carrier protein, they carry glucose molecules with them. This creates a concentration gradient of glucose, higher concentration of glucose in the epithelial cell than the blood. Therefore, glucose moves out of the epithelial cell by facilitated diffusion. This diagram, by the way, I've taken from the CGP A-level biology textbook. Absorption of amino acids. So amino acids are absorbed in exactly the same way, but everywhere you see glucose, so here or here or here, you would have. So protein digestion begins in the stomach with pepsin, and it's completed by a team of different enzymes in the lumen of the small intestines and the epithelium of the small intestine, which breaks polypeptides into small peptides into amino acids. Endopeptidase is an enzyme that breaks peptide bonds in the middle of a polypeptide. Exopeptidases are a type of protease which hydrolyzes the peptide bonds on the ends. So it hydrolyzes the end amino acids of the peptide molecules. Dipeptidase splits dipeptides into amino acids. So a summary of protein digestion. Pepsin is produced in the stomach, and it has an optimum of pH 2, and it is catalyzed by the enzyme pepsin, and it turns polypeptides into shorter polypeptides. Trypsin is released from the pancreas, and it has a pH optimum of about 8, and it functions in the duodenum, and it turns polypeptides into shorter polypeptides. Exopeptidase is found in the pancreas, produced in the pancreas, but it functions in the duodenum, and it catalyses shorter polypeptides to be hydrolyzed into amino acids by hydrolyzing that peptide bond. Dipeptidase is found in the epithelial cell membrane of the ileum, and it catalyses dipeptides to be turned into amino acids by hydrolyzing that peptide bond. Next bit is lipid digestion. One enzyme, lipase, hydrolyzes the ester bonds in the lipid molecules into glycerol and 3 fatty acids. Lipase comes from the pancreas and also the small intestine. Now there is a bit more detail to this. So actually the first thing that happens is bile salts will emulsify the lipids. Bile salts are just chemical, they're not an enzyme, which creates a larger surface area for lipase to act on. And then lipase catalyses the turning of lipids into monoglycerides and fatty acids. So now, absorption of the products of lipid digestion. Monoglycerides and fatty acids diffuse out of my cells in the lumen into the epithelial cell. Monoglycerides and fatty acids recombine with the triglycer to form triglycerides, which form globules which become coated with protein to form chylomicrons, and they leave via exocytosis and they then enter the lymphatic system. Assimilation. So carbohydrate assimilation at the liver, skeletal muscle, or at the liver or the skeletal muscle, glucose is stored as glycogen. This is hydrolyzed to provide glucose for respiration. Excess glucose is converted to triglycerides and stored in adipose tissue. Protein assimilation. Amino acids are used throughout the body to synthesize the proteins they may need. For example, enzymes, antibodies, hormones, transport proteins. And then excess amino acids are deaminated, releasing ammonia, then urea, removed by the kidneys. Lipid assimilation, transported as chylomicrons into lacteals, and then the body can also use the triglycerides for respiration by gluconeogenesis, or excess triglycerides can be stored in the adipose tissue. So amylase is produced in the salivary gland and pancreas. Site of action, if it's a salivary gland, it's in the mouth. If it's pancreas, it's in the duodenum. In the mouth, it's got an optimum pH of 7. In the duodenum, it's pH 8. Lipase is produced in the pancreas, works in the duodenum, and it's got a pH optimum of 8. Pepsin, found in the stomach, works in the stomach, it's got a pH optimum of 2. Trypsin is released from the pancreas into the duodenum, and it's got a pH optimum of 8. Next is control of blood glucose levels. So insulin is secreted by the beta islets of Langerhans in cells. At the liver, it causes the uptake of more glucose into the liver cells to be stored as glycogen. So that causes glycogenesis, which is the synthesis of glycogen from glucose. So it's turning glucose into glycogen. Glucagon is secreted by the alpha islets of Langerhans cells at the liver, and it causes glycogenolysis, which is lysis of glycogen into glucose. So the lysis or splitting of glycogen to release glucose. Or it can do gluconeogenesis, which is conversion of amino acids and glycerol into glucose. So gluco means glucose, neo means new, genesis means from the beginning or synthesizing. So it's making new glucose from things like glycerol and amino acids. Causes of diabetes mellitus, type 1 and 2. So type 1 is where the pancreas does not produce insulin. So the beta cells do not produce insulin. And the patient is dependent on insulin injections for survival or an insulin pump. Type 2 is insulin resistance, so a defective use of the insulin that is produced occurs. So the liver and muscle cells are no longer responsive to the insulin, and the patient is usually not dependent on insulin for survival. The treatment is usually first diet, monitoring diet and controlling your diet and exercise. Now looking at nutrition, the main food groups are fats, carbohydrates, vitamins, minerals and trace elements and proteins. So proteins are for growth and repair, obviously made up of amino acids. You've got many found in meat and other kind of sunflower seeds, nuts, etc. So nutrients are split into macronutrients, which are proteins, fats and carbohydrates, and micronutrients, which are vitamins, minerals and trace elements. And they help our body grow, maintain and repair all the cells and organs. So next bit is fats should make up less than 35% of our daily food intake. Provide a concentrated source of energy and insulation. Unsaturated fats are better for you than saturated fats. Essential fatty acids are omega-3 and omega-6, but we can't produce them, so we have to consume them. Carbohydrates, you've got simple and complex. Simple is sugars, complex is like starch, your polysaccharides. It should make up about 50% of our daily food intake, needed for immediate source of energy. Vitamins, water-soluble ones, you've got vitamin B, B1, B2, B3, B12, folic acid, B9, vitamin C, like ascorbic acid. They aren't often stored in the body, so they've got to be taken daily. Fat-soluble vitamins can be found in fatty foods like vitamin D and vitamin A, and also retinol. And then you've got minerals and trace elements. So trace elements like vitamin B and iodine, and minerals like magnesium, potassium, iron, calcium, help in various chemical reactions in our body, amongst other reasons. Exclusion diets. So these are prescribed for patients who have food intolerances or allergies. So lactose intolerance, you would exclude dairy. Calorie controlled diets. A calorie is the amount of energy it releases when used for respiration. The energy required for the body's essential activity that take place at rest is known as the basal metabolic rate, or BMR. To maintain a healthy weight, there needs to be a balance between the energy intake and the energy expenditure. So intake is from the things we eat, so energy from carbohydrates and fats. Expenditure is by physical activity, exercising, metabolic rate, etc. So it needs to be a balance between the two to maintain a healthy weight. If it's not balanced, this results in gaining or losing weight. So this is the principle behind calorie controlled diets. So if you want to gain weight, your intake has got to be higher than your expenditure. To lose weight, your expenditure has got to be higher than your intake. So assessing dietary and nutritional problems, you can calculate BMI using a person's height and weight. A normal BMI is regarded as between 18.5 and 25. So BMI sometimes is not the best tool when looking at professional sports people, because muscle does way more than fat.